everyone. Today we are learning about zebrafish behavioral test. So zebrafish is used in wide variety of diseases, and there are few diseases wherein the role of zebrafish is kind of more utilized in terms of behavioral analysis. For example, let's say neurodegenerative disorders. Okay, so neurodegenerative disorders demand the test linked to certain traits like memory, cognition, etc. So in this video, we'll be talking about what kind of test you can do with zebrafishes and what kind of apparatuses are used for that, and what is the analogy between the human and zebrafish behavior so that you can understand it better. So let's start. Let's first talk about what is zebrafish. So the use of zebrafish as a model organism began in year 1960. The scientific name of zebrafish is Teniorideo. It is a tropical fish native to Southeast Asia, and it is about 2.5 centimeter to 4 centimeter long. An adult zebrafish is 4 centimeter long, although you may have some zebrafishes which are slightly little bit longer than 4 centimeter. And in its larval stages it is transparent, and as it matures to an adult it develops these stripes that run along the length of the body and look blue in color. So right now on the right hand side of this screen you will see the black stripes which are running along the length of the body. Let me tell you, there is one more type of fish and you might confuse it for a zebrafish because it also have black stripes on it, but those black stripes move vertically along the body. They do not move horizontally in the body. So for zebrafish the lines move horizontally, and for the other one the lines are vertical, and that one is not a zebrafish. The picture in front of you is the actual picture of a zebrafish. So the females in zebrafish species, they tend to be less pink than the males and are fatter due to the eggs they carry. So when you will look at the fishes in your aquarium, you'll be able to make out very easily which ones are the females and which ones are the males, because females, they have a fatty body because they have eggs in it, plus they are less pinkish. So the male counterpart in this species is more pink, little bit shorter in size, but the female fishes, they're big and plus they carry eggs. Sometimes when you dissect them, you can actually see the clusters of eggs. So the complete genome sequence of the zebrafish was published in 2013 by an Indian lab, and its genome is 1505581,940 base pairs in length, and it contains approximately 26,247 protein coding genes. So this was about zebrafish. Now let's understand why use zebrafish in research. So number one is that they are small and robust, and they are cheaper to maintain than mice. And the break of daylight triggers mating in zebrafishes. So early in the morning is the time when they mate. So if somebody is working in the area of breeding them, they know when to collect the eggs. Zebrafishes, they produce hundreds of offsprings at weekly intervals, and this gives us an ample supply of embryos to study, because there are many scientists around, those who are working in the embryos of zebrafishes, because they are working on the developmental studies, and they grow at an extremely fast rate, developing as much as in a day as a human embryo develops in a month. And secondly, the zebrafish embryos are nearly transparent, so researchers can easily examine the development of the internal structures, and every blood vessel in living zebrafish embryo can be seen using just a low power microscope. So that is the awesome utility of why one should work with zebrafishes. Plus, zebrafishes have a similar genetic structure to humans, and if you don't know, they share 70% of the genes with us. Yes, 70%. Now, 84% of the genes are known to be associated with human diseases have a zebrafish counterpart. Isn't it awesome? We already have 70% genetic similar makeup, and out of those 70% similarity, 84% of the genes are known in human, and they have a counterpart in zebrafish, which are known to be, contributing in some or the other disease. This is wonderful. As a vertebrate, the zebrafish has the same major organs and tissues as humans. Their muscle, blood, kidney, and eyes share many features with human system. Zebrafish have unique ability to repair their heart muscle. For example, if the part of their heart is removed, they can grow it back in a matter of weeks. Imagine, this is just magic. And scientists are around the world working to find out the specific factors involved in this process to see if this, will help us to develop way of repairing the heart in humans, those who have heart failure or have suffered heart attacks. Isn't it so good? So, this is another dimension of why zebrafish should be used in research. The zebrafish genome has been fully sequenced to a very high quality, and this has enabled scientists to create mutations in more than 14,000 genes to study their function. Imagine the importance of zebrafish in research. Now, let's come to the topic which we are discussing today, why behavioral assessment is important in zebrafish. So we already know that the behavior of animal and human is metaphorically related to each other. Also, the study of animal behavior sheds light on the understanding of complex human emotions. So, in the quest for knowledge on human cognitive abilities and to understand the functioning of the central nervous system, animals have already been used as a prototype to show different behaviors when subjected to experiments on stress, chemical-induced diseases, cognitive impairment, environmental toxins, thirst, hunger, and many more. So, this is why the study of the animal behavior can help us understand the genes behind them, the logics behind them, and all that. So they have the tendency to remain in groups and they exhibit social behavior, which is known as shoaling. And we as humans, we like to be social all the time, isn't it? So if our experiments concerns wherein we have to look into how social they are, zebrafish is a very, very good model to be used. So thus, zebrafish has been used for mimicking the social behavior of humans, and studies have reported that the anxiety level and stress response can be measured in zebrafish by evaluating their diving patterns in a certain test which is called as tank diving test, and so forth, and another test as well, which we will see in a couple of minutes. So now, let's see what are the types of behavioral assessment. So primarily, I have categorized them in three parts. First is memory. So we can do tests which relate to memory and only memory. For example, where is my food? Where do I go to find my food? And all these things. And the other is cognition. Cognition is a wide variety term which takes care of many things. But if to put you, let me say that cognition talks about how to deal with people in general. I was talking about social tests, right? It comes in there. Second, if a new something comes to you, how you are going to respond. So your response is also a part of cognition, right? Now the third category is balance dysfunction, and I kept this as a category because it was neither going in memory, neither going in cognition. But if somebody is working with Parkinson's disease, they would really like to know the motor imbalances in their subject, right? So we do have tests to see this as well, which I'll be covering in a couple of minutes. So let's now see how to assess the memory of the zebrafish. So the first test which I have brought to you is Y maze test. So it looks like this, and your maze can look like many other things, but the thing remains same, that is the place where you are going to keep your fish. So there's a chamber where you keep your fish. So you have first gate, second gate, and third gate in this picture. So there is going to be a place wherein you are going to keep the food for your fish, and you are going to train your fish. So you will open this lid, your subject will come swimming from here. You will open this gate, the subject will go there, will not find the food, and you will keep on giving this training for months and months of time. And you At the same time, you will tell the subject that if you will go here, you are going to find the food the subject is looking for. So you have to train your subject for days, weeks, and months to go left and right, left and right, left and right. And you do this test with the subjects, with the zebrafish subjects whom you are going to put forward for a neurodegenerative disorder, for example Alzheimer's, Parkinson's, or whichever disease you are working on which concerns behavioral assessment, and specifically memory based behavioral assessments, right? So for days, weeks, and months you are going to train your subject. So later on, this subject right now is a control, but then you are going to give your subject certain treatments and it will become the model for any neurodegenerative disorder. And then when the subject, let's say, has Alzheimer's, you are going to put the subject here, but since you have given the subject training for whether to go left for the food or to go right for the food, so now the testing phase will come. So you'll keep on doing the testing on a regular basis, of course, but when the subject is really critically suffering from a neurodegenerative disorders, the subject will not be able to find where the food is, right? So Y maze is one of the tests which people use across the world to assess the memory of the zebrafishes. And what is the analogy with human behavior? See, you must have read the blogs, you must have seen the autobiographical videos, and otherwise also case studies also, that when a person is normal and healthy, they know what is kept where, right? You definitely know what is kept in your house at which place exactly. But once somebody starts having the dementic symptoms, they tend to forget things. So this is how this maze test gives us an analogy with the analogy of dementia in human subjects. So while you are making the model for, let's say, Alzheimer's, which talks about dementia, so this is how it is correlating with the dementia in humans. Now the next test in this series is native and new area test. So what you can do for this test is you can buy a chamber for yourself and you can create two different places in the same chamber. If you want, you can keep it the way I have presented it here. For example, you can keep the pebbles and the plants at the one side of the tub, and the other side can be left vacant. So while you bring your fishes from the vendor, so you must have kept those fishes in certain conditions, and you acclimatize your fishes for certain condition. So if you're keeping your fishes in this region, so your fish is accustomed for the pebbles, for the plants. But if you have kept your fish in a blank tank, then your fish is more accustomed and comfortable in a place which doesn't have a pebble. So this new native area test is a sort of a memory test for the fish, because now when you will come up with a chamber like this and you will leave your fish there, so if the fish comes from an area like this which has pebbles and plants in it, it will have the tendency of roaming more in this area. They will always go to the area where they are comfortable with. And isn't this the analogy with the human behavior? We tend, especially when we are weak, when we are when we are in a forgetful place, so we tend to go to place which we have seen earlier, which we sort of remember, instead of going to the place which we don't remember, right? So if your fish is healthy and controlled, they will remember the place from where they have come. But if your fish comes from a neurodegenerative disorders model, then you will find your fish moving at an equal rate in all of the places. I'm not saying that your fish is going to be seen more here. No, this is not the case. The fish will be seen at equal times, at equal places in the entire chamber. It will not have a preference for either native area or for the new area, because definitely our subject has now forgotten their native area. So you can expect them to be anywhere in the new native area test. Now, the second test is going to be from the cognition section. So let's see how to assess the cognition of the zebrafish. So the first test in this group is dark and light area. Let me first of all talk about the analogy with the human behavior. Okay, imagine that somebody kidnapped you, okay, and that somebody has kept you in a place which has an open area and which has a closed area, and they just slid you inside the room and slid you inside the the place, and they left. Okay, and you are definitely scared, and there is a place where you can hide and there is a place where you cannot hide and everybody can see you, and they have like camera reporting everywhere. Okay, and you know that everybody can see you, that where is the target right now. Where do you prefer to be at that point of time? Or the second example is, there is a thief in your house and probably might hurt you. Then where would you go? You would definitely go to a place which is dark so that you can hide yourself. Then now same goes for the fishes. While you're working with a neurodegenerative model again, the control behavior in this case would be that fish will always be found in the dark area. Do it yourself. You will understand. The fish is always seen at the bottom of the seabed. They are never flowing freely in the shallow waters, because what will happen if the fish is swimming freely in the shallow water? Anybody can come and pick them up and they're done, right? So they don't want that. So what they want is to be away from the danger, and to be away from the danger you will always see them in the dark and light area. So from my own experience, when we worked with the dark and light areas, we found our healthy fishes 100% of times in the dark area. But when you work with neurodegenerative models, you will find your fish going forth and here and forth and here and forth, from light area to dark area, light area to dark area, which tells us that their preference for the dark area has just gone. They don't know, because they do not sense fear. And sensing fear is a part of cognition. All right. So this is not a memory test. This is a cognitive test, where you would be when the thief when there is a danger lurking around. You will always be in the darker areas, right? So this was light and dark area, or dark and light area, whichever way you want to say it. The next is open field test. You might be, conversed with this test, because this is also done with rats. In fact, the light and dark test is also done with rats. So let me give you the analogy first with the human behavior. Same example, somebody kidnapped you and they had put you in a setup, and there are cameras everywhere, and let's say the guns are also there in the space, okay, if you can understand. So if there are guns there, and there is an order called as shoot at sight, okay, and you are stuck in a facility like this, would you be going here in the middle of the of that land, or would you be at the boundaries of the land so that somebody cannot see you from here? See, danger is from everywhere, but at least you would try to be in the place where nobody can see you, right? Like literally on the walls of that chamber, you don't want people to see you, so you will try to hide as much as possible, right? So what do you expect from a fish which is cognitively so intelligent? So a healthy fish will always be seen at the boundaries of the chamber. First reason is, since our chamber is made up of glass, so fish is thinking that it might be able to cross it. Fish doesn't know it is glass, and fish doesn't know that they cannot cross it, but they will at least try, right? So what they do is, they come with the full force and they butt their head on the glass, and this is called as head butting. So head butting is also a feature of a very healthy normal fish. They try to escape. They butt their head on the walls of the apparatus, and this is how they know that they understand. They can actually see you. So while you are performing the behavioral test, it is always advised that you monitor them from a distance. please do not move around and around your apparatus. Do not make sharp noises in the lab because they sense everything, and then it is going to hamper with your results. Your results will be erroneous at the end of the day. So now let's get back to the open field test. As a human, you will never be seen in the middle of the field, right? You will always at the boundaries of the field wherein you can like stay safe. So similarly, the fish will be seen at the boundaries. But same neurodegenerative model, if you're working with a neurodegenerative model or a dementia model, you will see your fish swimming here and there with no preference for any place, right? So this is what open field test is. Next is novel object test. So let me give you the analogy with human behavior first of all. If somebody captured you and then after some time they kept a bag in front of you, you're already scared because you are not in your aquarium. They captured you, hijacked you, and then they have put you in this in this box, and now they are turning up with a shiny bag and you don't know what to do. What will you do? As an cognitively sound individual, you will never touch the bag. We don't know whether the bag has bomb or whatever, right? So we do not want to go near anything which can hurt us, right? So that's the human behavior. But how does fishes behave around novel objects? So I have like two brilliant things to tell you from my own experience. First is, you will always find the fish staying absolutely away from the novel object. They will never ever come closer to that object. But that is if your test is ranging from 10 to 20 minutes. If so, and in some cases they do not swim at all. They do not swim at all because they are in shock because of the administration of the object. So now the trick here is, step number 1 is you have to put your fish in the vessel, and step number 2 is introduce new object. If you have already kept the object prior to the introduction of the fish in the in the test, then your fish will not be scared of that object because that was there when I came. That was already there, so I'm sure that is the part of the environment. There is nothing to be worried about, right? But if you have put your fish for the very first time and then after 1 minute or 2 minutes you are putting it in the water and the fish is sensing it that something new has come, then it will show this behavior. It will not swim at all for as long as 20 minutes. I have seen it. And even if it is swimming, it will always be very rapid swimming. It's like this, and it will never ever come across. It will never come into the proximity of the new object. But if you will leave your fish in this chamber for like 2 hours, you will see your fish, going and checking it out and all the things. So this particular test does comes with a timeline. Okay, it does comes with a timeline, and from my experience, 20 minutes is enough. After that, fish also gets bored because the object is not moving. Right? So the subject doesn't feel that it is a predator. Predator always moves. All right. So if in place of this, if you would have brought some fish who can eat zebrafish, so the fish will never rest, will keep on running here and there. But since the object is not moving, because the object is not giving any chill to this subject right now, it gave the subject a chill when it was introduced, but after that it understood that it is a non-living object, number 1. Number 2, it's not moving at all. That means it is not a problem for me, right? So this test does come with a timeline, and the timeline here is like 10 to 20 minutes. Okay, so this is what I wanted to tell you. Maintain the timeline, plus introduce the new object when you have already put the fish in the vessel. Otherwise the fish will never understand it as a new object. So this is something from my own experience. I hope it helps. Another is social preference test. So this is another study which we were doing in our lab, which comprised of, testing how the fishes behave when they are away from their peers. So what I did was I kept few fishes in separate vessels for months. And when I had put them together with the other fishes, I found that they were not going close to any of the fishes. They were not going close to any of the fishes, and they were rather confused, and they did not go back to them for at least 30 minutes. So yeah, this test also comes with the timeline, So after 30 minutes or 40 minutes, you will see your fishy gelling up with the other fishies, and then it's just gone. You'll not be make out where your fishy is, okay? But otherwise, zebrafishes are such a social beings that they like to be always in clusters, okay? So if you have kept any fish isolated for a long time, it will have a difficult time it will have a difficult time communicating with other peers. And if you will see the same analogy with humans, you will find it very correct. If there is a child who was always kept at home and did not go out, see the buddies of his or her age group, you will find them having a hard time talking to the other children, and otherwise also, if there are people, those who are in mental asylum, they're now fine, or somebody who is in depression and stayed alone for a very long time, you will not find them chit chatting with somebody, and even if you are sitting on their face, they will not look at you, okay? Because they cannot talk, because they don't know how to behave now, because they were socially isolated for a very long time. And zebrafish was a brilliant model. So if you are learning something about social preferences or doing something in this area, this is an excellent model. This is an excellent model. So if you're working with a dementic model, for example, neurodegenerative disorders model, you will see that your fish is just fine. If somebody is coming, meeting, it's okay. If somebody is not coming, it's okay. It will go move around everywhere. You will see that the fish is not taking part in the shoaling movements or something. It's just freely flowing everywhere. So as far as the neurodegenerative model goes, they flow freely everywhere. You just cannot make out anything from them. So while I was working, I was actually confused, because I had some healthy controls which were kept in isolation, and then there was this neurodegenerative model, and when I put them separately in a social preference test, it was very difficult for me to pinpoint the differences between them. So even the healthy fishes which are kept in isolation for a very long time, they also show certain symptoms. You feel maybe they are dementic, so this test is a failure when it comes to distinguishing between the Alzheimer's and the socially impaired fish, and a socially impaired fish because of the isolation. So yes, that eventually helped my team to come up with a new type of test, which I'll cover in another video. So yeah, so this is one of the tests which we use for assessing the cognition in the fishes. So now let's talk about a very specific test for assessing the balance dysfunction of zebrafish. So first I have kept in this category is tank diving test. Actually, this test should be there in the cognition section also, but I was not planning to keep it at both places. So you can imagine that this is from cognition section also, and you can put it in this section also, which is we are trying to see how the fish is maintaining its motor balance. So we were working with a neurodegenerative model again, and the model was for Parkinson's disease. So we saw that this tank diving test was giving us more than just the cognition, which I'll show you in the form of a video also, which we just created for this PPT. So we had to create a different setup, because obviously we cannot keep the actual videos here for the presentation. So we just created another tank for you to see. So now let's see. This is a tank, and this tank is filled with water. You can see this is the line, and this is the level of the water. This level is called as upper region, this is medium region, and this is bottom region of the tank. So what you have to do is you have to divide your tank into 3 regions, upper, medium and bottom, and you have to decide this upper, medium and bottom from the line from where your water starts, because you really want to cut your water into 3 layers, 3 equal layers. Okay, so I was just telling you, in case of cognition, where would you find your fish to be? Will it be in the will it be in the shallow waters, or will it be in the depths of the water, bottom of the bed? So it will always be in the bottom of the bed. Okay, now let's learn about the analogy with the human behavior. So definitely, if you if you are in a danger sort of situation, so you would want to hide yourself and go away from the danger, right? So you will always find yourself in the area which is like safe for you, and the area safe for the fish is like bottom region. So at least our subject is thinking that we are not going to harm them. Okay, so you will always find them in the bottom region or in the medium region, but the healthy control fish will never ever come in the upper region, given if you are standing there. If you are watching them from a distance, so for a very long time, like they'll be seen in the medium and the bottom region, but at some point of time they'll come in the upper region. Okay, but if you are they'll come in the upper region only when you're watching them from the distance. If you are standing there, they will never come on the upper region. Okay, so this is the test for the cognition. But now let me tell you how it is useful if you are working with some disease which can cause motor imbalance, for example like Parkinson's disease. Okay, so this is a dummy video which was created for this particular lecture. So let's see. So you will see in this video that the subject was not able to swim even. Okay, so let us show it to you. Okay, so now the subject has gone, and you will see the subject was slowly, freely falling and was really unable to swim. Although the subject will swim eventually, but this is what we saw in Parkinson's disease. And there is another thing also which I want you to see. So when they are in vessels, this is how the disturbed motor features in the Parkinson's model are seen. You can see that the fish is not able to maintain its body balance, right? So while you're working with the rats, there are different sort of things you can see in them, but while you're working with zebrafishes, this is an equally good model. It is an equally good model, and as far as Parkinson's and Alzheimer's go, they are like very, very good. Thank you for watching. Please like and subscribe, and I'll be putting more videos for you to see on zebrafishes. If you want me to do some particular video on zebrafishes, please write me in the comment section and I'll come up with the video. Thank you, and I hope it was beneficial for you.